So welcome everyone to this special virtual via Zoom speaker presentation on behalf of the National Capital Area Chapter to the Depression and Bipolar Support Alliance. National DBSA is celebrating its 40th anniversary this year. There'll be a special celebratory program on the weekend at the end of September in Chicago, where it is headquartered. And we are honored tonight to have a virtual speaker presentation. We've over the years tried to have presentations by doctors, other professionals in the field of topics of interest to those with mood disorders. And tonight we have Dr. Mark Kvarta of the National Institute of Mental Health. His talk, as our previous talk, and probably a couple of others to come, is arranged by our medical advisor, our new for the past year medical advisor, Dr. Carlos Zarate of the National Institute of Mental Health. I think it's correct that Dr. Kvarta works with Dr. Zarate. Let's see. I think we've hit the 7 o'clock hour. I hope a few more people will be joining us. Dr. Kvarta, you can find information on him on the both on the NIMH, National Institute of Health webpage about him, and on the University of Maryland Medical School. He has bachelor's from Cornell, PhD in neuroscience from the University of Maryland in Baltimore, and MD from the University of Maryland School of Medicine. And he's been at the University of Maryland Medical Center, Shepherd Pratt Hospital, physician training program he was in the general psychiatry residence training. Let me read his bio sketch. My translational research focuses on how stress affects our neurobiological framework to understand key symptoms of depression and other serious mental illness. I focus on linking neuroimaging, neuroendocrine, and genetic measures to clinical measures of depression and stress in humans. I aim to increase our knowledge how exactly stress across the lifetime changes our brains to cause specific symptoms and ultimately to create a path for developing and understanding better treatments. And from the National NIMH website, Dr. Kvarta is the director of the Outpatient Mood Disorders Clinic and chief of stress pathophysiology research in the experimental therapeutics and pathophysiology branch of the National Institute of Mental Health. He received his undergraduate degree magna cum laude from Cornell. He earned degrees at Maryland. I mentioned his residency. And he's been joined the faculty of the University of Maryland School of Medicine as clinical associate professor of psychiatry and physiology, establishing a neuroimaging lab focused on stress pathophysiology, genetics, and circuit biomarkers of serious mental illness within the neuroimaging research program at the University of Maryland Psychiatric Research Center. He joined the ETPV program at NIH in 2023, certified in psychiatry by the American Board of Psychiatry and Neurology. Okay, and there's a more detailed selection. He has numerous publications which you can visit online. So he's got a very distinguished record, and he's speaking today on the next generation of antidepressants, ketamine, and what are the three letters?
HNK. So let me
HNK.
bring out my title slide, make sure everybody can see that.
Please go ahead. So let me, without further ado, let's welcome Dr. Kvarta. We look forward to his talk, and we will, after he goes through it, we'll take questions or comments. Thank you.
Perfect. Great. Alvin, thank you so much for that wonderful introduction.
Okay.
Yeah, we have a lot of, especially in government, we have a lot of very long titles, right? So we love our acronyms.
Yes.
And I guess, DBSA is not too different. You have your acronyms too.
Mhm.
All right, so I'm delighted to be here. So as Alvin mentions, I'm director of our outpatient mood disorders clinic at National Institute of Mental Health over in Bethesda. And our branch is the experimental therapeutics and pathophysiology branch. We focus on mood disorders in particular. So experimental therapeutics part is we're trying to develop new treatments, and the pathophysiology part is we're trying to understand what went wrong in the first place, whether that's at the cellular level or sort of brain wide. And of course, yeah, Dr. Zarate is my boss, my chief. So he recruited me from University of Maryland, where I had happily been for many years, as you could tell from my bio. So the focus of today's talk, even though I love understanding about how stress influences the brain and can make symptoms worse, I'm going to focus today on ketamine as the prototypical fast-acting antidepressant, how we think about it, and hydroxynorketamine, or HNK as we call it, and yet another acronym, as the next generation of antidepressants. So of course, I'm easy to find online. If you have any questions that you don't have time to ask here or think of later, feel free to email me. I'd be happy to answer those. And I just want to give credit to a couple of more famous people that I've borrowed slides from. So Dr. Bhatti at Stanford and Dr. Nemiroff at University of Texas. All right. So just a quick disclosure. I am an employee of National Institutes of Health, Department of Health and Human Services, United States government. I do own a patent, but I'm not gonna discuss that here, and I've assigned the rights of it to University of Maryland. And any views expressed here are my own and don't represent the United States government in any capacity. All right. So the general outline here. Going to go through a few statistics about depression. You're probably familiar with it broadly and how to impact society, but I'm always shocked by how pervasive it is. I'll talk a little bit about the common treatment pathways, probably the things you're most familiar with, as well as some of their limitations. I'm going to discuss the unique challenges of treatment-resistant depression, sometimes called difficult to treat depression. I'll get into some of the best and newest tools out there that help, a quick survey of a few things, as well as ketamine, of course, and then get into the journey of ketamine and its inspiration of a new generation of treatments. And we're hoping HNK is one of those. And of course, time for questions at the end. All right, so a little bit about depression statistics. So, worldwide, it's one of the top 10 causes of disability, morbidity, and mortality, just life years lost, productive life years lost, whether that means disability or early death. Over 10% of individuals suffer from a mood disorder each year. And mood disorders we consider major depression, dysthymia, and bipolar disorder. So those are the mood disorders in general. And of course, both major depression and bipolar depression can have treatment-resistant depression. So that means depression that doesn't seem to get better with adequate treatment. So about a third of cases of mood disorders have treatment-resistant depression. I try not to put too many words on every slide, but sometimes it's unavoidable. There's about 1 million deaths per year from suicide worldwide. And the most visible changes in depression are problems in mood, behavior. So meaning you're not getting um, your your typical interests, you're not engaging with things, you're not doing your hobbies, you're not interacting with your family the same way. Changes in your circadian rhythm, so changes in your sleep and eating habits, and then your overall functioning, what we call functional impairment, which would be your performance at work or in school, and fulfilling sort of your responsibilities. And I'll get into a little bit more specifics about how we diagnose later. So a little bit more about overall prevalence. So here is from a recent year. So this is 2021. And this is just looking at the overall prevalence by percent of the whole population by each demographic here. So overall, about 8% of people will experience a depressive episode in a given year. 2021 was a fairly typical year in terms of the statistics. So that's why this one's on here. So overall, about 8% of people will have a major depressive episode. So that how we define a major depressive episode is basically depression lasting for at least 2 weeks, meeting most of the symptoms, and I'll discuss the symptoms later. A little bit higher for females over males. And then if you look at the age, it's actually the biggest problem in our transitional age. So we have our 18 to 25-year-olds, almost 20% in a given year. So these are our college students. These are people just starting their lives, right? But it certainly doesn't go away in any decade of life, and 20s, 30s, 40s, 50s, and beyond. By race and ethnicity, tends to be highest amongst Native Americans, whites, Hispanics, and actually highest for those who identify or report having 2 or more races or ethnicities. So it's the highest rate of depression. Okay. There we go. Now just, we looked at the 18 and 25-year-olds and those rates were pretty high. What if we go back a few more years? So here's just looking at our adolescents. So really the rate of depression kicks off at the preteen years and through adolescence. So you see 12 to 13 years old, it's about 13%. And then if you can read these numbers, by 16, 17-year-old, more than a quarter, more than 25%. So this is, generally a red alert for our adolescents and keeping a close eye on them. And even higher for females yet again, right? Almost 30% of young ladies in this age range. And then finally, again, people who identify with 2 or more races, again, more than 25%. So they'll have the highest rates, especially in this age group. So one thing I like to discuss briefly is how is clinical depression different from ordinary, or what we say subclinical fluctuations in mood? What's just having the blues or having a couple of bad days versus having depression? So in the normal course of human events, right, we should mostly be in this green area if you don't have a major depressive episode. If it goes down below that, that would be considered a depressive episode. If it goes up higher than that, that's the other side. So that's a manic episode. And if it goes just a little above that, that'll be a hypomanic episode. And each of these has their own specific criteria by the Diagnosis and Statistical Manual, the DSM, now version 5. But in a nutshell, a depressive episode would be at least 2 weeks of having most of the symptoms of depression, most of the days. And a manic episode would be having the symptoms of mania for at least 7 days. And that's elevated mood, sometimes grandiosity, sometimes pressured speech or racing thoughts, that kind of thing. But focusing today on depression. So that would be in this red zone here. All right, so a little bit more specific on who meets the criteria for major depressive disorder. So I've been talking about depression kind of broadly, but I did point out that there's you can have depression different flavors. So either major depressive disorder, you can have a depressive episode as part of bipolar disorder. You can have a depressive episode due to medical conditions. And you can also have sort of lower levels of depression or more rapid cycling depression in dysthymia or other disorders. But today we're going to focus on major depressive disorder. So in a given year, about 3.8% of the population will meet the criteria here. And you need to have at least one of these two key symptoms. So the first one is sadness or depressed mood. So it'd be, severe case of the blues. And then the other is what we call anhedonia. So you're not being hedonic, right? You're not enjoying things. So it's a loss of interest or apathy. So in this little cartoon, the bunny's not enjoying the carrot. He's just ignoring it. So you need to have at least one of these. Sometimes you have both. And then you need to have at least 4 of these other 7 symptoms. So that'd be changes in weight or appetite, sleep disturbances, usually insomnia, but you have many flavors of that, right? Difficulty falling asleep, difficulty staying asleep, waking up too early, changes in psychomotor levels. So that would be psychomotor agitation or retardation. So moving or thinking or speaking too fast or too slow. Overall fatigue, of course, beyond just what you would get from the insomnia. Guilt or worthlessness. Executive dysfunction. So usually it's concentration difficulties. Sometimes people tend to ruminate a whole lot about the most negative events, either from the recent past or more distant past. And that can be pretty impairing. And in the worst cases, suicidal ideation. And the most catastrophic cases could lead to a suicide attempt. But sometimes people just struggle with thoughts of death or suicide for many, many years and never even have a suicide attempt. Is my overall volume okay? Is everyone okay? Good. Okay, so you need at least 4 of these, but it's kind of mix and match, right? And you need at least one of these 2 core ones, but you can have both of these and 3 of these. You can have just one of these and 5 of these. There's many, many different ways to kind of mix and match. So the scientific word for that is heterogeneity. We have many different things that we're calling one disorder. So that can lead to a little bit of difficulty. There's a lot of differences in how we do this. So there's actually hundreds of ways to meet the criteria for major depressive disorder. So that's something to keep in mind too. You've probably heard this quote used for other things, but if you've seen one patient with major depressive disorder, you've seen one patient with major depressive disorder. So, there are some things in common amongst many people, but everybody is their own unique story. So as I mentioned, hundreds of ways. And actually, if you take into account, a lot of these have multiple options even within them. You actually get into over 100,000 different ways to meet the criteria, right? Because you have guilt or worthlessness. You can have increased appetite or decreased appetite. Those both kind of count. So you can see that all the mixing and matching, it's pretty massive. And so overall, as I kind of mentioned before, depression manifests as problems of mood, behavior, circadian rhythms, and activity levels with impairment in functioning. And of course, functioning, as I said before, doing everything you want to do in your life. All right. So how do we usually treat major depression and other depressive disorders? So a little tongue in cheek. Here's the candy dish. It says Prozac. I don't own this. So here's what a typical treatment algorithm might look like. So it's broken down into 4 stages. You start with stage 1. You've been dealing with symptoms of depression. You've gone to see maybe your primary care. Maybe you've been referred to a psychiatrist. But if you're seeking medication as opposed to therapy, you start with stage 1 here. And so if you look all the way on the right, you might see a couple familiar drug names. But this would be a typical first pass. So it would be a single medication, so monotherapy, and then you would try to increase the dose if you don't have a response to it at first. And so there's the reuptake inhibitors. So these are things you might have heard SSRIs. So this is things like your serotonin reuptake inhibitors. So fluoxetine or Prozac, Lexapro, Celexa, things like that. You have other ones, mirtazapine, or the trade name is Remeron. There's tricyclic antidepressants, amitriptyline, vortioxetine or Trintellix, trazodone, which is also a sleep agent, and so on. So typically you'll start with one of these in stage 1. You'll try one, and then you'll increase the dose if you can. Now, of course, that took probably at least 3 months to get into the psychiatrist's office, take the medication, give it a couple months to work, because that's really how long some of these take. Before you kind of say, well, it's not working, or it's not working enough. So then you'll go on to stage 2, what we call augmentation or switching. So augmentation will be adding a second thing. And so there's recommended things to add on to something you're already taking, or you'll switch. You'll switch to a different one here, or switch to a different class. And then you can also switch instead of to another medication to something like ECT or electroconvulsive therapy or electroshock therapy, sometimes it's called, or TMS, so transcranial magnetic stimulations. That's using a magnet to stimulate parts of your brain, kind of a little less intense than ECT. And these do have shown to be pretty effective in some populations, but not completely, not for everybody. Otherwise, we'd start there. If that doesn't work, we'll go to stage 3. So that we'll move on to, if you haven't done ECT yet, you might think about that. You might try some of these older medications, monoamine oxidase inhibitors. They're a little bit more difficult to tolerate, so we don't really go there right away. And you'll see ketamine's listed here. Typically in the community, you can go to an infusion clinic and get IV infusions a few times a week. There's a intranasal spray that's now been approved of S-ketamine. It's called Spravato. I'll mention that again later. And there are these online shops where you can get ketamine lozenges that you have mail ordered, and you take them online while your therapist watches you. So that's a little bit less regulated compared to the standard, which is infusions. And then stage 4, experimental. You might repeat other stages. Some people try literally everything on here on the way down, right? So some people will try 20, 30 different things. And we see them at the NIH Clinical Center. You might try a clinical trial. You might try other medications that aren't quite approved yet, but might have some scientific basis for adding them on. So, of course, as time goes on, this might be a year at least, maybe more if you've tried multiple things here. So, in summary, try a drug or 2 or 3 or 4, try neuromodulation maybe, and then try experimental treatment, maybe a clinical trial. And of course, I changed the axis here, so instead of treatment time, it just says a long time. All right, and then this is illustrated here maybe a little differently. So here's in the green, here's time along the bottom here as we go along those 4 stages. And here's what we hope for patient improvement. So the optimal response is the first medication does the trick. You go from your low mood here all the way to basically cured, right? In remission, as we say. For somebody who doesn't respond till the second stage, you follow this one here. Got a little better, but didn't get all the way there. We have a line here showing what we consider a response based on the numbers versus remission, fully remitted, feeling completely euthymic or normal. No symptoms of depression anymore. The third stage we might have, here's the first trial, maybe got a little better, second trial, third trial, fourth trial, fifth trial, maybe finally figured out, the right cocktail, as some people say, if it was a certain combination of medications. And then, of course, some people, just nothing ever seems to really work that good. They never really make it to what we would consider a clinical response. So this is really where we do our work. All right, so why the need for different drugs or interventions? So I think you can probably guess based on the last few slides, there's no one thing that works for everybody. If there was, we would just do that. So the biggest trial is called the STAR-D trial, the Sequenced Treatment Alternatives to Relieve Depression. This is the largest study looking at something like this. So they looked at about 5,000 patients who had come in for a first round of treatment, either to their primary care or some community psychiatry offices. So they'd never been on treatment before, and they all met the criteria for major depressive disorder. And so what they did here was they basically put things head to head against each other for these 4,000 to 5,000 patients. And so there was a first round where they just compared some of those medications, and in the second round you could either switch or add something to that first round medications. And for people who still didn't get better, they went to a third round and a fourth round of either switching or adding things. And the remarkable thing is that in each of these 4 rounds, the overall response rate was about 30% each step of the way. So no matter what you started with, you had maybe a 30% chance of reaching that remission standpoint after that first treatment. So there's no overall best treatment at any of those stages. And then at the second stage, whether you switch medications or add something, again, maybe about a 30% response to those people remaining, right? So if you're only getting 30% each step of the way, you're always going to have people left over. So sorry, I'm just going to move the little box here. It's in the way for me. It's not in the way for you. So overall, about 30% of people actually got completely better and met the remission criteria. So 2 out of 3 had some response, 1 out of 3 met full remission, but also 1 out of 3 remained ill, didn't meet the criteria for either response or remission after 4 steps. And of course, those 4 steps, as I mentioned before, takes about a year, right? We think about like 3 months per adequate trial for these traditional medications. So after a year, 1 out of 3 have remained with severe depression. All right. So what is treatment-resistant depression? So we're gonna get into it a little bit here. You might recognize this guy. That's what I think about sometimes. Also called difficult to treat depression. Depending on which paper you're looking at and exactly how you define it, but I think you're getting the picture. Anywhere between a third to a half of people don't have a adequate response to 2 antidepressants, meaning their symptoms don't improve. So this is the criteria that we use to say you have treatment-resistant depression as opposed to you just have major depressive disorder. You have not responded to 2 adequate antidepressants. So that means taking it for long enough to for us to say it's adequate, and also that it's an adequate dose. 33% don't respond after 4 antidepressant trials. And then some of that's from the data I already showed. So as I mentioned, standard drugs take up to 8 weeks. And then some people are just unable to tolerate due to side effects. Every body is different. Everybody's body is different. And treatment-resistant depression leads to adverse health outcomes overall. So in addition to lost productivity and everything you do in your life, withdrawal from your family and social engagements, that kind of thing, there actually are higher rates of metabolic disorder, cardiovascular disease, stroke, things like that. So, untreated TRD actually leads to shorter lives. And this is completely not even considering the fact that there's about 40,000 suicides per year in the US. Okay. So here's a different way to look at the course. So euthymic again means normal mood or sort of neutrally happy. Over time, as your mood goes down, now you meet the criteria for depression. And so this is again sort of time, but no specific time course. Kind of mentioned all of that already. There is a time lag. So let's say down here is when you decide to go see your psychiatrist or even your primary care and get prescribed their first antidepressant. Let's say you get better from your first treatment. Now we have a lag of onset 10 to 14 weeks until you start to get that improvement. So there's a big time period here where you're suffering. For our hopes for the next generation of antidepressants and anti-suicidal ideation treatment, we're looking for rapid onset. So within hours or even maybe a day or two, you start feeling better and you get back to your routine. And I'm sort of jumping the gun here, but I'm going to talk a lot about ketamine as the prototypical drug that does that. Okay, and a couple more factors associated with increased risk for treatment-resistant depression in particular. So amongst everybody who has depression, here are the factors that might predict less likely to respond to those first 2 standard treatments. So age, more chronic depression, so many more years untreated, high levels of anxiety and comorbidity with anxiety, being more socially isolated, higher body mass index, suicidal ideation is a bad prognostic factor, a family history of suicidal ideation. There seems to be some genetic component there, as again, a family history of alcohol use disorder. And again, this seems to be more of a genetic component than an environmental effect. Okay, so just very briefly, this is a figure from the former director of the NIH, Tom Insel. For about 15 years ago, there was an effort to, instead of just looking at things based on the definition from the DSM, so here we have major depressive disorder, here we have dysthymia or mild depression, here we have bipolar disorder, to try to integrate them all and look at common factors within the whole group, whether it's genetic risk, brain activity, specific symptoms, specific physiology or features of life. And using that, we can actually recategorize people. Some of these people have more in common across disorders than within a disorder. And so that's where it's called research domain criteria, or RDoC approach. And so this is trying to move beyond just the stratification by a diagnosis. So you may have seen that clinical trials over the last 15 years in psychiatry have taken this approach more and been a little bit less strict about who they're recruiting, as long as you have some of the symptoms they're focusing on. So how do we? Sorry, I'm moving the little box again. So how do we actually design the next drug? Here's what a typical pharmaceutical company might do, trying to get from an idea to a new drug on the market. It costs on average about $1.8 billion, including the failures on average per drug they've tried. And so this is a funnel. Here's discovery research. These are your ideas. This might be things that are being publicly funded, maybe being done at universities, not necessarily being driven by a drug company yet, but doing some of the legwork to advance major ideas. Then things, this might actually be useful for depression. Maybe this was a neuroscience lab that was just trying to understand how brain cells work, and somebody had an idea, this might work for Alzheimer's, this might work for depression, this might work for bipolar disorder. So now it'll be in preclinical development. Maybe a drug company is picking it up. Maybe the university is trying to push it forward. Maybe us at the NIH are trying to push it forward. So we'll have preclinical development. Then we'll go to phase 1 trials, which is really that'll be the first time it's in humans. And usually it's not in patients, but in people that might not have the condition you're looking for, but just to make sure it's safe. And you might do some things to show that it's doing some of the things you hope it'll do for patients. And then you'll go into phase 2, which will be in patients, and you'll be looking for both the efficacy as well as safety. Then move on to phase 3, which is really these larger trials. You might have hundreds of thousands of patients where you're getting the treatment against whatever the standard of care is. Then assuming this all goes well, you can register your drug with the FDA and get approval. And so this area, it's called the Valley of Death, is because from here to here, about only 10% of discovery research gets to these early phases, and only about 10% of those move on to phase 2. And then maybe 10% go from phase 2 to phase 3, right? So there's less than 1% of these ideas getting fed into the funnel make it out through here. So that is a lot of ideas, a lot of work, a lot of money, a lot of time that goes into trying to figure out what treatments will get better. So given all of this, how do we even think about designing new treatments that'll be more effective than what the past 50 plus years have given us? Here's the overall clinical approval success rate for something that started a phase 1 trial and then got to approval. And you see CNS is central nervous system. So that would include both neurology and psychiatry. You see less than 10%. And that's just from, this middle part to approval. All right. And of course, I mentioned the heterogeneity of depression. If one drug works for somebody's suicidal ideation, it might not work for somebody else's insomnia and low mood. So the complexity of diseases, the human brain is remarkable. It's hard for our brains to understand itself. There's a lot of philosophy around that, but basically just trying to figure out exactly what the brain is doing, right? There's billions of nerve cells, and there's trillions of connections between them. Limited segregation of patients into biological levels of trying to actually design what they're doing. You have a lot of problems just in terms of regulatory hurdles, things like that can come up late. And of course, if it takes too long, another company can make a generic. You're no longer under your patent protection. So if you spend all this money trying to get through here, if you take too long, you might not be under your patent protection anymore. Someone else can make the same or similar drug and sell it for cheaper. So there's a lot of risk for companies. So anyway, how do we even think about designing next treatment? So part of it is relying on that early science, that discovery science. And luckily, we have learned a few things over the last 50 plus years. So what is happening to our brain cells? And I'm going to be very, very brief with this part, but just to say we do have some direction with this based on our research. So the previous and the current generation has relied pretty heavily on animal models. And the current generation and the next generation are using some animal models, but also starting to leverage computational models, AI, machine learning, all those things, and as well as non-animal models like stem cell derived neuron models that we can grow in a dish. And so what have we learned from that? So here's an example of these are neurons that are derived from the frontal cortex. So this is the big part of the front of your brain that is responsible for what we call executive function. It's an area that other mammals don't always have. We think a lot of our reasoning comes from there, as well as some mood regulation. And it turns out this area is deeply affected by depression. So in a lot of our models, we find that chronic stress actually does a lot of the same things that we see in people who have been chronically depressed. And so you can see a control. Here's a neuron in a non-stress condition, and this is just a few micrometers long. The actual length might be up to a millimeter, depending on how long some of these branches are, but the actual cell body is only about 10 micrometers, so it's a thousandth of a millimeter. And all these little things, looks like something you pulled out of your garden. All these little branches here are things that connect to other neurons. So that's where the 100 billion neurons becomes several trillion connections between them. And you can see after the chronic stress condition, there's way fewer branches, right? If this was growing in your garden, you'd want this, right? It's healthy, it's growing great, whether it's a tree or something, a vegetable or whatever it might be. Whereas this one, you can see there's way fewer branches, and they're just less complex, they're less long, and there's fewer of them. So this is what chronic stress does to our brains. And we see this in depression. If you zoom way in, each of these little bumps here, so this is zoomed way in, right? So this is just a single branch maybe here. Each little bump, that's one place that a connection can be formed with another neuron. So that's where our processing power comes from, right? You think about the processing power of your computer, and that increases year after year every time you get a new computer. But if in the depressed condition and the chronic stress condition, you can see there's just a few of these bumps, right? So now you've just downgraded your computer. All right, and so the bottom line is depression can impact the brain, and we can see it under a microscope. There's actually physical changes that happen. And of course, I have to put in something for stress because I spent most of my career studying stress as well. Too much stress itself can impact the brain too. It can be toxic. And so what does this mean? So the connections between the neurons in depression are weaker, but they can be strengthened. So here's some old work from mine. You can actually record the electrical signals between brain cells. They communicate through electricity. Again, I'll use the computer analogy, just like our computer circuit board does. And you can measure the electrical current that's passing inside the cell. So this is just, you don't have to worry about that, but a specific type of excitation that goes into the cell. And if you look at the black, this is in the sort of the euthymic or the happy condition. After either chronic stress or in the depressed condition, you see that that deflection for the black trace here is much smaller. So this is less electrical current that the cell can pass in communication with another cell. And then finally, here's SAD plus successful treatment. So we're in the happy state again. There's actually a recovery of that transmission. And in animal models, this only happens in those that were treated and also had a recovery of their behavior. So there are a few things we can monitor to see if they're sort of exhibiting depression-like signs. So even if you treat them and if they don't get better, this reversal doesn't happen. So we're pretty confident in the way that some of these brain areas are working in depression. Let's see. So in this particular example, we used an SSRI. So this was actually Prozac or fluoxetine in the hippocampus, which is another key brain area along with the frontal cortex that seems to be affected in depression. And the hippocampus, a funny word, it's basically Latin for seahorse because of its shape, but it's important for learning and memory. And so you can see some of the cognitive effects of depression are located in the hippocampus. So this is in vitro, this is in a dish, but the oldest way scientifically we have of strengthening of synapses or the connections between these brain cells is through electrical stimulation. So here we use the drug, but you can do it with electrical stimulation in the dish. And you may have heard of the first one that we do clinically. So this is a form of neuromodulation, or modulating the way the neurons are communicating. But ECT is sort of the prototypical one. So you use electricity delivered with electrodes to induce a seizure briefly, usually less than a minute. And so the seizure activity in the brain actually improves mood. And so this is all tightly controlled. It's nothing like it looks like that you might have seen in the movies. And some people completely swear by this. They've tried 30 different treatments, medications, they have side effects, or just nothing worked. And they go for ECT treatment after a few visits. And like, I haven't felt like this in 20 years. And so some people will swear by it and just stick with it forever. Part of our group is actually experimenting with using lower doses of electricity to have fewer side effects or less of a seizure and still have some of the clinical response. So that's sort of hot off the press. This is actually some other work from others in our group. It just shows if this is a depression score, you came in here with severe major depression. After 4 treatments or so, it's cut in half. After 12 or so, you probably don't meet the criteria for depression anymore. And these are pretty big samples. This is hundreds of people that have come in for treatment that have not responded to other things. So they have treatment-resistant depression. So ECT is kind of been around for a while. A newer version of neuromodulation is called accelerated theta burst stimulation. And of course, we have another acronym, and the Stanford protocol, the Stanford Neuromodulation Therapy, or SAINT. Basically what they do here, so I mentioned TMS before, which is using a magnet to induce electrical activity in your brain, does not cause a seizure, but can strengthen synapses in our models as well as in patients. And so what's special about this new therapy is that it can use this more mild stimulation to the brain than ECT, but it's inspired by your own brain imaging. So people will go into the MRI, we'll see where activity is deficient between some of these key brain areas. Again, here's the frontal cortex, the PFC, and the communication between them. And so based on the activity in these brain areas, we can say, we're going to position this coil a little bit further down to stimulate this area, or we'll position it a little bit further back to stimulate this area, because it seems like that's where things are different. So this is a really exciting development over the last few years that speaks to that heterogeneity, right? Everybody's different. So this is almost a personalized medicine approach, right? To treat your depression. And so we've started doing that as well as part of our standard of care. So I do see there is one question.
Yes. So I was wondering, you mentioned how, like in the Saint protocol, out of curiosity, that what they do is they measure like the communication between the brain regions,
Mhm.
and you said that they use an MRI, because I would have thought they would use an fMRI for that more.
Yes, exactly. You are correct. Yep.
Okay.
So it's the same machine, but you use a different protocol to make it do fMRI record imaging instead of structural MRI. But yes, great point. Yes, I may have oversimplified that. Okay, and then just very briefly, something else that's out there you may have seen a little bit about, mushrooms, psilocybin. This is kind of taken off. There is some evidence in treatment-resistant depression. So you can see, again, here's sort of the depression score. People do these sessions over a few weeks and show a pretty good response to their overall depression score compared to a control group. Of course, these are very different. I took the slide out, which shows these dosing sessions usually have 2 chaperones with you. You'll spend 6 to 8 hours while you're doing this dosing session in a very comfortable environment. A weighted blanket. You might have nice smelling things, soft lighting, soft music, that kind of thing. So it's very hard to model this around the placebo effect. But even trials that are starting to design better placebo controls are still showing pretty good responses with this. Okay, so that's just, people always ask about that, so I just threw that in there. All right, so I'm going to get into ketamine, which is the prototypical one. And this is, I'm not going to belabor the details here, but this is just to show that we've kind of been on this path of developing ketamine for many years. So originally synthesized in the 60s as a dissociative anesthetic. Approved by the FDA as the buddy drug and administered to soldiers in the Vietnam War in the 70s. A lot more work on it in the 90s, and just some era of psychedelic therapy of 70s through 90s with traditional psychedelics or with ketamine was kind of being explored around that time in different settings. It's just some cool art. The ketamine tiger that represented this person's experience with ketamine. And traditionally ketamine outside of treatment has either been used as a anesthetic, a tranquilizer, especially in veterinary purposes like horses, and abused as a recreational drug. But those are all at much higher doses than those we use. So the development of this class of drugs as potential fast-acting antidepressants wasn't until 2000. So this drug had been around, but been used at mostly higher doses and not in treatment-resistant depression specifically. So that was the first few observations started right around 2000. Incidentally, the predecessor of my current program was the mood and anxiety disorders program that came around in the 90s. So we were, not me, but we were there in 2000 to leverage these findings. So there's ketamine. This is the first bag in the first clinical trial that Dr. Zarate used. So I stole this picture from him. So the pivotal trial in 2006, ketamine as a rapidly acting antidepressant for TRD specifically, was done in 2006 on our unit. And then we had multiple replications at IRP, which is our program, intramural research program at NIH in Bethesda, over the years. Independent replications at multiple sites. And these are all important things for getting your drug approved. And then finally in 2019, there was the approval of the intranasal spray version of S-ketamine developed by Johnson & Johnson, which is just S-ketamine, so it's a little different than ketamine altogether in infusion. But this was one of the first approved fast-acting antidepressants. Okay, so for the rest of the talk, I'm just going to talk about what we do at the Experimental Therapeutics and Pathophysiology branch. The short version is just ETPV, rolls right off the tongue. Here's our unit. It's a very nice psychiatry unit. It is a locked unit because it meets standards for a psychiatry unit. And we do lots of cool stuff. So this is a magnetoencephalograph. So it uses magnets to look at brain activity. So it's a little different than fMRI. There's not many of them. Don't worry too much about this. There's not going to be a quiz. But the overall mission of our branch is to develop novel and rapid-acting pharmacological therapeutics for patients with severe mood disorders, especially treatment-resistant. And step one is understanding what's happening at the molecular level, moving on to proof of concept studies, and then ultimately better treatment. So this is Dr. Zarate, my boss and your medical advisor for your chapter. Okay, and so here's some of the data on ketamine. This is going back to that 2006 study. Individuals received either a single infusion of ketamine or a placebo, and there's their depression score. And you can see within minutes, there's actually an improvement in symptoms, and especially at 4 hours, especially at day 1 and day 2. And there was still a statistical effect at day 7, so up to a week from a single infusion of ketamine. And these are all individuals who did not respond to traditional medications. And so you can see here the proportion of participants responding at different time points, right? So at 1 day, almost 71% of individuals responded, compared to the SSRIs or other classical antidepressants. Takes at least 8 weeks, and this is kind of giving a little too much credit, 60 to 62%. All right, and so what we do, we look at many different things. We mentioned the MRI. We can do MRS, which is looking at specific molecules in specific brain areas. So like glutamate is a neurotransmitter. We can look at other things. The MEG, which this is a similar picture. fMRI and structural MRI, which we mentioned. So other studies, we can do sleep EEG, right? So this is electrical activity in your brain while you're sleeping. And we have found important things that happen for people who respond to ketamine there. And then, of course, a lot of molecular studies, either that we do in our lab or that we do with collaborators across the country and around the world. And of course, we're getting more and more into genetics. We can look at polygenic risk scores based on other data sets. we actually have a collaboration with NIAID, so that's the National Institute of Allergy and Infectious Disease. They've been doing whole genome sequencing on all of our patients who wish for it to be done, and we haven't been able to really dig into the data that much yet, but I'm really excited. We have over 300 people that actually have agreed to do this, and a really rich data is going to be coming out of that. All right. I'm just checking the time. I want to make sure I leave time for questions. But there is a convergence of these different things that might be rapid acting antidepressants. So here's psilocybin, or the magic mushrooms. Here's a different compound that's been approved for postpartum depression. And then we have ketamine up here. So there is overlap in the pharmacodynamic mechanisms, but I'm not going to get into the details here due to time. But basically, in a nutshell, downstream from the actual receptors that they're targeting off the bat, there are convergent cascades that are acting on the proteins that are translated based on your DNA transcription. Neurotrophins, things like BDNF, or brain-derived neurotrophic factor, changes in the configuration of how those neurons look. So think back to those pictures of the your garden with those brain cells, and actually neurogenesis, so actually the growth of new neurons. So all these rapid treatments all seem to converge there. Okay, so I kind of said this is a little bit of a different way. We try to go from the molecular and cellular level up to the systems level up to clinical trials, and we do all three things at our branch. And our eventual goal is more data-driven precision medicine. So this again harkens back to the RDAC approach, but we're collecting so much data across so many modalities with deep phenotyping, integrating different levels of data, and then applying new computational approaches to hopefully leverage precision medicine. Say, you might be good for ECT, you might be good for ketamine, you might be good for HNK. All right, and so I'm going to skip that one just due to time, but again, here's another model about ketamine. So I mentioned the brain cells before. If you zoom way in and look at the little bumps, these are dendritic spines, they're called, and these are where the connections are formed between different brain cells. So again, this is similar to the black and white picture I showed you before from real neurons. This is a cartoon version, but at baseline, there's a certain number of these spines. The ones in red are stress sensitive, so after stress, they're going to be plucked away. The cells actually reabsorb them. And there might be an immune role as well, so immune cells have some role here. And then after ketamine, just within hours, we have some of these come back. And in different models, people can track even the same branches of these neurons over time through treatment. Okay. And so what have we learned? This is probably way too much, but we've gone from just a few things on this schematic. This is a connection between two neurons. This might be the spine from the previous figure. We've gone from just seeing the neurotransmitter and maybe one receptor to all of the things. And each of these things in the pink boxes are different drugs under different stages of development as fast-acting antidepressant. So I'm not going to go into it. There's too many and decades of science there, but just to give an overview. So just like many drugs that we take, aspirin, other things, our body metabolizes what we take pretty quickly into other things. And so ketamine is broken down into norketamines and hydroxynorketamines. And if you're a chemist, you probably understand what each of these, addition of an H or an OH is, but basically it's our livers generally that break these things down. And so an incidental finding we had with some collaborators from University of Maryland is that this metabolite, the HNK, actually this one, seems to have a lot of the antidepressant effects, but does not have the sort of dissociative side effects that ketamine altogether has. So it might be different metabolites, or ketamine itself does. And also this one seems to stick around a lot longer. And so that has led to us developing this as a candidate drug for treatment-resistant depression. And so here, again, here's the time scale. Here's the comparison to monoamine drugs like SSRIs. If you do have any side effects, some people don't, but if you feel a little loopy or have a little bit of dissociation from this low dose of ketamine, it usually ends within 2 hours. Let's see if I have the right animation here. There we go. But the HNK sticks around for 3 to 7 days. So initial drug is long gone at 4 hours. The dissociation ends at 4 hours, but people's mood lasts for up to 7 days, and this drug sticks around for up to 7 days. And so these are some of the observations that have led us to develop this. All right, and so I'm going to skip over that, but here's a kind of a snapshot of what our clinical trial looks like. So folks that come in with treatment-resistant depression, they failed. We're a little bit more lenient, just failed at least one past medication. They have a depression score that's pretty high. They'll come onto our unit. Sometimes we can do this outpatient as well. So if folks know people in the area that might be interested, we can do this outpatient, at least the first phase. And so in phase 1, people come in, we'll do a full assessment. You'll come off your medication. Depending on what medication is, sometimes we can do it pretty quickly. Sometimes, stretch it out to 5 weeks or so to make sure you don't have side effects. Then you're supposed to be completely off of medications for 2 weeks. And then you receive either the HNK or placebo saline. And these are infusions just like we would do with ketamine. After 4 infusions over 2 weeks of a escalating dose, you'll be drug-free for 2 weeks. So if there was HNK, we want to make sure there's no carryover effect. If it was placebo, we don't want you to know that. And we don't know it either. Only our pharmacist knows. And then you'll move on to research condition 2, which would be you'll cross over to the other condition. And that way we have a control within subject's control, which is what we call it, as if somebody responded to placebo a little bit, we want to make sure that the response to the actual study drug was greater. And then once people complete their study with us, we offer standard care treatment to get you back onto what you could get in the community. Or sometimes be able to try something else, right? So at stage 1, 2, 3, 4 before with different medication combos, we might be able to have some ideas for things that other folks might not have had. And so you can see all the different time points we are doing ratings. We're doing biomarker research. So that's the MRI, the MEG that I mentioned, the MRS, as well as other optional procedures. So we actually have a new sub-study launching about comorbid pain. HNK might be effective for that too. All right, so just in a nutshell, I kind of said all this already, but could be up to 11, 12 weeks if you do the whole study. And most of it would be inpatient, but we can do parts outpatient. And so this is our design for double-blind randomized controlled trial. Okay, and this is just to say a lot of stuff goes into it, but most importantly, we do need volunteers. All right, so that's it. I do want to acknowledge, it takes a village and it takes a city, and it takes really a nation. And so all of our collaborators, both at the NIH and outside, we have patient advocates on the unit. We have our pharmacists. We have our trainees that come through and come up with new ideas. We have our staff, and of course, inpatient, outpatient clinical staff. And I just want to acknowledge my boss. And I started to list out all the people we have, but we have over 75 people in our group. These things are very complicated. We need people to help us with the regulatory submissions, patient safety, nursing, everything, our laboratory, that kind of thing. So that's it. I do want to thank all the patients and their family and our community partners, like DBSA, like NAMI. We love working with everyone. So I do want to say thank you for listening. I'll bring this slide back up. It has our main email, but again, you can search me pretty easily, and hopefully Alvin will distribute my email address, or if it wasn't on the notice. But I just want to end with this. One of my heroes, Robin Williams, and everyone you meet is fighting a battle you know nothing about, so be kind always. And so with that, I'll take any questions you might have.
Thank you, Dr. Kvarta, for an excellent talk.
Thank you.
Covering a lot of ground. I hope we've learned some things. And let's throw it up to questions. People can put questions in the chat if they prefer. If you want to come online and ask it, you can indicate. Just speak up. Don't know if anyone's watching. For anyone who may do an electronic hand raise, you can just unmute and talk.
Yeah, I don't see any hands raised.
I believe Jake had a question.
Oh, that's right. Jake, did you have a question? Well, we'll come back to Jake. Kristen.
Sure. So thank you for the talk. It was super helpful. I have two questions actually. But I also don't want to monopolize. So let me just start with the first one, and then if other people have questions, I'll back off. The first one is you mentioned that in the clinical trial, you're doing some work with comorbidity around pain. Have you found any data to suggest that people with bipolar disorder, major depression, have a higher level of experience with chronic physical pain than the general population?
Yeah, I know for sure major depression, yes. I don't know for bipolar disorder in particular. So we're working with experts. We have a pain center at NIH, and so we're launching this new sub-study. Dr. Lauren Atlas, you can look her up as kind of the expert in the comorbid pain area here that we're working with. But the answer is yeah, certainly yes for major depression.
Okay, excellent. As I suspected. And then the second question I have is so interesting to learn that there may be a link to genetics research. So actually in my job, I work with scientists who do genetics research around blinding diseases.
Oh, wow.
And that's a big, that's a big part of it because a lot of, especially rare blinding diseases are very clearly linked to genetics. But I wasn't aware of bipolar disorder or major depression having any commonly known clear link to specific genes. Is there data to suggest that?
Yeah, there are a few. I mean, so the coarsest way to look at it is just overall heritability. So there are some sort of more famous the twin studies, especially if twins were raised in separate households, right? Because you have to try to tease apart, what's a shared influence of the environment or their upbringing, that kind of thing. And so there is a a double digits heritability of major depression just from genetic basis. It's actually a little bit lower than other psychiatric diseases. Complex ones, so schizophrenia and bipolar have much higher genetic risk. And so for depression, going beyond the heritability, looking at specific genes, there are these big data approaches. So 23andMe has been offering their data to researchers. Of course, everything's de-identified. There's a psychiatric genomics consortium, PGC. The UK Biobank is a really big effort to track people longitudinally, mostly middle-aged folks, but they do get some genetic data. And so we can calculate things like polygenic risk scores based on the pattern of all the different gene, all the different alleles you have at each gene site. Of course, there is a commercially available thing called GeneSight. So you send in a gene sample, and they use their proprietary calculation based on your genetics to say whether or not you might be a good candidate for different antidepressants, right? So you might be a fast metabolizer, you might be a slow metabolizer, you might be more likely to have side effects from this class of drugs. And that's based mostly on the genes that make up all the proteins in your liver, right? So the way that we kind of metabolize drugs. But there's other things to it too. Beyond that, there have been, we've had more success looking at whole pathways of molecules that interact as opposed to a specific gene. So, there have been about 100, 150 individual genes identified. But if we look at the whole pathway, it works a little bit better. So things like stress processing pathways, there's a few inflammatory pathways that light up, a couple of thyroid binding proteins light up, and that might not be surprising too for anybody who's had troubles with thyroid. So there, we are making progress in that area, but there's no one gene that says this gene says you'll get depression. So they each seem to have a 2 to 3% effect size, right? So there's no one predictor. So it's pretty complicated, and you might predict that based on the heterogeneity of the symptoms that I kind of went through.
Okay, that's helpful.
Yeah, good
Okay,
question.
thank you.
There are a couple questions in chat. Gary asks, I've started taking Spravato. Is there a typical frequency of administration? how long could I safely continue to take it?
So the frequency of administration is typically 2 to 3 times a week. And of course, ask your doctor that you're working with, what kind of makes sense for you. There is a sort of standard pathway, and then assuming you've had some response, people tend to either stop completely or reduce the frequency. And some of this is based on what, kind of what your doctor orders. I know there's some clinics that will kind of space you out and say, you were doing it twice a week, you're doing it 3 times a week. Now let's do it once a week. Now we'll do it once every other week. Now we'll do it once a month. And that's sort of your maintenance level. Since it's still relatively new, there's not really a strong direction on guidance of exactly what everyone should do. So it's a little bit more trial and error on that front. What's working for you and what you can kind of still do. It is a little time consuming, as you probably know, if you're going to a clinic to do it. You kind of have to be monitored for a couple hours, and I think you're supposed to have a chaperone to get you home. So logistically, it can be a little difficult. And I see Jake was able to unmute and has been waiting a while.
Oh, Jake, go ahead.
Yes, thank you. I'm wondering whether there have been any studies, or actually whether any drugs are developed or attempting to be developed that sort of duplicate the effect one might receive physically from massage therapy or sex or gambling.
Sort of the things that are more euphoric.
Exactly.
Right. So yeah, so we think a lot about these things in terms of, I mentioned anhedonia, right? So the inability to have hedonistic sensation, sort of the classical definition, but not enjoying life, right? The things you should enjoy. So the counter example that you pointed out, the things you enjoy a lot. So we do think a lot about what things really drive up the reward processing. And so classically, you think about like the dopamine reward pathway, but there's some brain areas in particular that we think about. So the ventral tegmental area and the nucleus accumbens are sort of the dopamine pathway, and they send dopamine out into the rest of your brain that's processing things. So there's sort of coarse drugs that do some of that very broadly throughout the brain, but so far they might not always be the best thing. So amphetamines do that, cocaine does that, but we want safer ways to do that, more targeted ways to do that. So there are more nuanced approaches to do that, sort of in animal models with genetic procedures and that kind of thing. But in terms of drugs, none of them have really made it past that valley of death that I mentioned in terms of drug development. Good question. Thank you.
Okay. And Kevin says, I was hospitalized.
I can read it. It's
Oh,
fine.
go ahead, Kevin.
Okay.
please.
I was hospitalized from October 2008 to March 2009 at NIMH, and I participated in the double-blind longitudinal study where one infusion of ketamine was placebo and the other infusion was active. So my question, of course, is how different have ketamine protocols changed in the past about 20 years?
Yeah, good question. And thank you for volunteering. I mean, you were here before I was. That's great. I've only been in NIH for a few years. Yeah, good question. So the standard protocol is a little bit similar to what I described with the S-ketamine, the nasal spray. So typically it would be 2 to 3 infusions per week, usually up to 4 weeks, and that will be the induction period. And then you'll either stop and just rely on a conventional antidepressant if you're feeling much better, or if you feel like you need the maintenance, you kind of space it out to once every other week, once a month, some people. And of course, the ketamine infusion is not FDA approved, so it's still used off-label. So unfortunately, that means insurance doesn't really cover it. So if you don't happen to have a few $1,000s extra a week to pay out of pocket, trying to do the nasal spray that'll be approved by insurance is kind of the way to go. There are plenty of infusion clinics around that will do it sort of cash only, but it is fairly expensive. So people will often do it and then be limited by how much money you have to dump into it. Typically about $500 an infusion if you're paying out of pocket. And of course, as you know, coming to NIH, we don't charge for anything. We actually compensate people for their time participating with us. Gordon.
Yes, thank you, thank you. So, I actually, I have, just disclosure here, I have bipolar, but I also have a movement disorder that's considered to be a neuropsychiatric because it's a functional movement disorder.
Mm.
And I remember, this whole discussion with the TMS and then the intermittent theta burst protocol, it reminded me of this study that was going on at the NIH under the PI was Dr. Cho, and I think because of funding cuts, I was told ultimately the study didn't, they were only able to finish phase 2, but they weren't able to progress further than that. Do you know, like, I guess this is less of um, a mood disorder thing, but I guess more interdisciplinary in the sense crossing between mood disorder for me and my functional movement disorder, because I guess there's a non-trivial overlap. If there would be any way to find out what the results of that stage 2 sort of study were, and then B, if there would be a way to sort of, get that sort of treatment. I mean, I know that there's like the right to try law, but that only applies to people with terminal illness, not like what I have. So would the other option be just like going to another country to try to see if, and another trial that would be doing something similar, or finding a TMS clinic that would be willing to employ that protocol, if that's even legal? I'm curious, just your brain there.
You might be able to find a clinic that would be willing to do it and still kind of, I don't exactly know what was it the Saint protocol or the AI TBS protocol? I mean, there are clinics around that are doing it as standard of care. You can do that.
Yeah.
In terms of the results question, so any clinical trial is supposed to, I believe the guideline is within 1 year, the results need to be posted to clinicaltrials.gov. So the overall results should be there. I mean, there's no individual data or anything like that, but I would keep an eye on that and see if you can find your specific trial you participated in.
And do you think that specifically, like, so it uses the intermittent theta burst, but what they specifically do, and I think this is why, I alluded to the fMRI earlier, in one of my earlier questions, was because they specifically, like based on the abstract I read on clinicaltrials.gov, like some number of months ago, they like use the fMRI to specifically target certain parts of the brain. I don't entirely understand that process, but I'm guessing that would probably be only able to be done at an academic institution like, Stanford or the NIH, where they have like an fMRI and all of that high-tech equipment in the first place, right?
Yeah, I mean, just the TMS machine itself is pretty expensive, so there's not a lot of sort of community providers that offer it. There are some folks in the area. I keep a list of places that are offering. I haven't started a list for the AI TBS in particular, but places that are doing TMS and are more academically oriented that might be open to it. So if you want to shoot me an email, I might be able to send you a list.
Thanks, Mark. Appreciate it.
Yeah, thank you so much for your question. And then Jake, did you have another?
Yes, I do. In your talk, you referred to depression as a mood disorder. I've always thought of it as an emotional disorder. Do you see any distinction between the two?
I do, and we do spend a lot of time discussing and debating and certainly thinking about the words that categorize people's life experience. And so I think just for communication's sake, mood disorder is kind of the catch-all for, we throw in depression, bipolar, dysthymia, other things kind of in with it. So I think you're right in terms of depression. You certainly think a lot more about the emotion and the affect, AFFECT, affect. I prefer affective disorder, which I guess kind of goes along the same way. So it is a good question. And I don't have a good answer for you other than write to the DSM and state your case.
Thank you.
Yeah, thank you.
Other questions? Let me ask you, are there likely to be increases in FDA approval of these treatments?
Let's see. So I know there's been a lot of advocating to try to get the ketamine infusions themselves approved.
Yeah.
unfortunately, it kind of, I showed the money involved. There's sort of generic, it's a very cheap drug to make. It's not necessarily protected under any patent anymore. So finding the right person, or the right company that has the money to kind of push it through the whole FDA approval process hasn't really been there. So I don't want to get too much into that because I'm not certainly not an expert in the FDA side of things. But there are sort of existing barriers in terms of an activation energy to get it to move.
Okay. On the genetic question, there is at least a test that people with difficult to treat mood disorders can get to do it, get an individual genetic test of what may be more or less likely to work. Is there likely to be more coming on that sort of thing?
Probably, as we kind of learn more, especially, we start applying machine learning and AI and that kind of thing. I think we're going to keep seeing more patterns that weren't necessarily seen with the first generation of those tests. There's a comment, Caroline, another term I've heard is energy disorder. Do you have any thoughts about that?
Oh,
yes. I mean, that's a really interesting point. So sort of my gut reaction to that is thinking about the psychomotor changes, right? So usually people with pretty severe depression but have that psychomotor retardation, it's really quite profound, and it's a little bit rare. You don't see it very much, but it just looks like people, the person is moving in molasses and they're talking much slower, and all their family members will say, I've never seen them talking this slow before. And that's why I started to think about that Eeyore picture. Yeah, just every sentence just takes way more for their thoughts to come out, and it's hard for me to do that because I talk really fast. I'm from New Jersey, so it's a little difficult. But it's just noticeable if you know somebody ahead of time, and then they start going through that. So certainly for that energy disorder, and then fatigue. Just the fatigue, the insomnia, and the low motivation to even get out of bed and go about your day. Oh, it's going to take too much energy to make myself a meal. I'm just going to grab this bag of chips, right? So you can see how these things kind of all play into together. It's going to be too much energy to make it to see my friends, or to make it to that church function, or just to get myself ready for the day. So yeah, I do like that as well. And then Kristen made another point, energy characterization, and then yeah, pain is big too. Thank you for sharing.
other questions or comments? Thank you for putting this information on the last slide of how to participate in clinical research. We should make a point of noting on our website.
We're very easy to find. If you just Google this, you'll find us.
Good. We'll try to make sure it goes on our site or in our weekly newsletter. Let me put in a plug. Our website is dbsanca.org. And if you go there, you can find, well, a number of things of community resources, including the NIMH institution we're fortunate enough to have in our area. And we put out a weekly newsletter that you can see. You go on the website, but you can also click on it to enter your email address and receive such each week, usually on Sunday. Also note that we have a variety of peer support groups, and there are links for other family support groups, but we have, I think, 10 peer support groups, 7 of them meeting virtually. One of them is generally on Thursday evenings. Tonight's an exception, but generally it's each Thursday, and there are, I want to say, 7 other, a few specialized, others general peer support groups, and we now have 3 in-person groups meeting weekly in DC and Silver Spring, and our newest is in Ashburn, Virginia. Okay, does anyone else want to say anything else or ask anything else of Dr. Kvarta?
I just want to say thank you so much for being so engaged, asking such great questions. Thank you so much for your attention. Oh, Jake, you got
Okay, this
thumbs
program,
up.
by the way, has been recorded, and this talk and slides will be put up on the website, on our chapter website, and it'll be noted in the newsletter when that's available. So feel free to come back and check on things, and Dr. Kvarta has indicated you can send him email questions. Anyone want to say anything else? This was certainly an excellent talk. But some people got to comment, and we'll look forward to having more such programs.
Thank you so much for having me. This has been delightful. Thank you.
Thank
Thank
you.
you, Dr.
Have a
Kvarta.
good
Thank you,
night.
Doctor.
Night.
Good night, all. Okay. If there's nothing else, I guess Stephanie, you're the host, so it's for you to officially end the meeting. Thank you all for joining us.
All right. Thank you so much.
Good night.
Night.